Okay friends, so now today we will going to discuss about Haemophilus influenza. Okay, so this video is just an introductory video, so where we will going to discuss about the basic characteristics of Haemophilus influenza. Okay, so let's get started. So Haemophilus influenza, first of all, is a small. It is gram negative and coccobacillus. Okay, so coccobacillus means its shape is in between round for coccus and the linear for bacillus. Okay, that means the shape is in between round and linear. Okay, so that means in this position. Okay, so now which can normally colonize in the human respiratory tract. Okay, that means human Haemophilus influenza will cause respiratory illness, right? So Haemophilus influenza has a thin peptidoglycan layer, and it is obvious because we have discussed that it is gram negative bacteria, and I hope you know that gram negative bacteria possess a thin peptidoglycan layer along with a outer membrane, right? So as it having thin peptidoglycan layer, it is a gram negative bacteria. Okay, that means it cannot retain the crystal violet, instead it will color pink due to the safranin, right? So our Haemophilus influenza is non-motile and it is facultative anaerobe. Okay, that means it can survive in both aerobic and anaerobic condition, and it is our catalase and oxidase positive. Okay, so we have learned about that our Haemophilus influenza is small, it is gram negative, and the shape is coccobacillus, right? That means in between round and linear. Okay, so Haemophilus influenza is a type of bacteria which stains pink due to the safranin dye, right? And it is non-motile and facultative anaerobe, and it is catalase and oxidase positive. That means Haemophilus influenza have the ability to produce catalase and oxidase enzyme. Okay, and it can grow in the chocolate agar media, because chocolate agar media contain two necessary nutrients for the survival of the Haemophilus influenza. One is the factor 10, which is also known as hemin, and the factor 5, which is nicotinamide adenine nucleotide. Okay, our NAD. So it can also culture in blood agar, but with a special condition. That means we will use blood agar containing Staphylococcus aureus colony. Okay, so here the Haemophilus influenza can get its necessary nutrient by hemolysis of the red blood cell. Okay, so in both the cases it will give a grey, convex, smooth or transparent colony. Okay, so the colony morphology is convex, smooth, grey or transparent. Okay, you have to remember, and it can grow in blood agar along with Staphylococcus aureus and in chocolate agar alone. Okay, chocolate agar provides both the necessary nutrient factor 10 and factor 5, and blood agar along with Staphylococcus aureus produces factor 5, right, by hemolysis of the red blood cell. Now we are going to classify the Haemophilus influenza. So we are classifying Haemophilus influenza into two parts. One is encapsulated strain, another one is non-encapsulated strain. Okay, encapsulated strain means it contains the capsule. Okay, and the encapsulated strain can be divided into 6 serotypes depending on the capsular antigen present on the surface of the capsule. Okay, that means Haemophilus influenza is surrounded by capsule in encapsulated strain, and in encapsulated strain there can be presence of 6 different capsular antigens. Okay, that's are A, B, C, D, E, F. Okay, among them the B strain is important because it causes infection to human, and the uncapsulated strain, which devoid of polysaccharide capsules, is important. Okay, because it also have the ability to cause disease. So that is all about the basic introduction of Haemophilus influenza. So we have discussed that Haemophilus influenza is a small, Haemophilus influenza is a gram negative bacteria, it is a coccobacillus. As it is gram negative, that means it is clear that it is having a thin peptidoglycan layer and outer membrane, right, and it stains pink due to the safranin, right, and it is non-motile and facultative anaerobe. and it can easily colonize the respiratory tract. That means it will going to cause respiratory illness. We will going to discuss about the symptoms of Haemophilus influenza in upcoming videos. And it can grow in both the blood agar and chocolate agar media. Okay, but in case of blood agar, it also requires staphylococcal aureus colony for their growth, right? It requires factor 10 and factor 5 for its growth. Okay, and in both the cases, Haemophilus influenza appears as a gray, transparent, and our convex colonies, or in sometime it also gave smooth colonies. Okay, that means convex, smooth, gray, or transparent colonies. Haemophilus influenza can be divided into two parts. One is chocolate agar media. Chocolate agar provides both the necessary nutrient. One is factor 10, another one is factor 5, and another one is blood agar with staphylococcal aureus. And we have divided Haemophilus influenza on the basis of presence of capsule or not. So one is encapsulated strain, another one is non-encapsulated strain. Encapsulated strain can be divided into 6 different serotypes from A to E and F, right? And among them B is important because it is causing the disease, and uncapsulated strain responsible for the causing disease, although it's devoid of capsular antigen. We're going to discuss in our upcoming video how those encapsulated strain and unencapsulated strain causing disease. Okay, so this is the introductory video. So we will again come back with another video where we will going to discuss about the virulence factors. Okay, that means the weapon of the Haemophilus influenza that helps Haemophilus influenza to attack and then destroy and evade the host defense mechanism. Okay, so till then, bye bye.